Hello and welcome, Mindsetters. I know that you have just tuned in to Learn Extra Live Grade 10 show. Today is Life Science with Cheryl, and we have some exciting stuff today, guys. Call all your friends right this minute because today we are doing practicals. We're doing different kinds and also the theory that comes with the practicals. So make sure that today you are at your best because it is going to be such an awesome show. You guys know how to get hold of us. Our Facebook page is facebook.com/LearnExtra and our Twitter handle is @LearnExtra. Guys, send in any questions that you may have. Some of you on the page are already saying that is my favorite subject. You die for the subject. Your name is Katleho, same as my name. Awesome. So I'm looking forward to you guys today. And if you have joined us, guys, let me know on the page that you are here because I love chatting with you guys. Boy, I have missed you so much. Over to you, Cheryl.
Thank you, Katleho. Good evening, guys. Welcome. Today, what I'm going to focus on first, I want you to just have a look at the setup in front of me, is we're going to look at the food test practicals. I know that at the beginning of the year, all right, when you did biochemistry, I think they just skimmed over food tests, but I want to do the actual food test for you today because it's a really important stepping stone. Because I know in the textbooks they say to you, this is what you must do and this is the color that must happen, but I don't think you'll actually have a picture in your mind until you've actually seen it or done it yourself. And also, as you know, that in our new CAPS document, practicals are going to form part, or should be forming part, of your paper at the end of the year. So you're going to write paper 1, paper 2, and then you're going to write a practical paper, which that is actually the fun part. All right? But the reason why I wanted to do the food test today is they form the basis of lots of the practicals that you might be doing later on. So from now all the way to when you get to matric. So what we have learned in the food practicals, this is something that you must never forget, please. When we test for glucose, when we test for starch, when we test for protein, right, when we test for fats, the results there don't only necessarily tell us what is in the different foods, but it's going to have a far-reaching consequence, as I'm going to explain to you now. If you have a look at the setup, remember when we do a practical, all right, it is going to involve having apparatus in front of you. I have set up already just to try and, all right, get a little bit of the momentum going so that I don't have to sit and mess and whatnot and have to clean up already. I think I've stained the desk, all right? So I've just set up for you, but it's very important that you need to know, all right? What are we doing? We're doing a practical. For each thing that we do, what do I need to do? I need to know an aim. So what am I testing for? So when I did the food test isn't just one test. It was a variety of tests. If you have a look in test tube A over here, all right, my aim is to test for glucose. So I am going to see if glucose is present in this test tube. B and C over here are going to be, all right, testing for lipids. Now, I've put 2 here because one I'm going to show you what happens when I just put water in, and the other one what happens when I put the reagent in. And the last one over here, this test tube, is going to be to test for protein. So look at my aim, all right, what am I going to do to test for glucose, to test for lipids, to test for proteins, and over here you'll see on this tile, all right, I've got a piece of bread there, and that is going to be for testing for starch. So those are my aims, the 4 things. I've got 4 different food types, if you can use that, and what I'm going to do is I'm going to see if glucose, fat, protein, and starch are present in that. Now, as you will have a look, if I set up, all right, here's my experiment. I've put my aim. Now what do I need to do? I need to have an apparatus check. I have test tubes, test tube racks, I have beakers, I have all my reagents, which I'm going to get to you now. I've got a water bath. I've got everything that I need in front of me, right? So that's my apparatus. Then, after I've set up, what must I do? I need to go into the method. Right? For each test, I need to be able to set it up, point step by step, and hopefully in the right order, because if not, then things can go a little bit haywire. Okay, let's start first of all with the glucose test. Now, in here, you'll see what I have put, and now this is very important. Listen to what I talk when I say about the reagent. Some people might use the word universal indicator. A reagent is a chemical substance that will change color if what I'm looking for is present. Okay, so now this is blue. Okay, and what it is Benedict's solution. Right, so when I'm going to test for glucose, my reagent is going to be Benedict's solution. Some of you in your school, sometimes you can use the alternative, which is Fellings A and Fellings B, right? Two separate ones that you combine together, and that is also blue. Now, so the color is blue. Now what am I going to do? I want to see if glucose is present. Here in this beaker, I have put a little bit of grape juice. I actually wanted to get a little bit of glucose, but I did forget. And I'm just going to put a little bit of glucose in. Okay? And now what I need to do is I need to speed up the reaction. So I'm going to take this, and as I have prepared over here, all right, I am now going to put it into a water bath. What am I expecting? I am expecting a color change. If glucose is present, the blue will change to orange. Okay, the blue will change to an orange. And we'll put that in there for a while, and then we'll come back to see if it works or not. All right, so there we go, water bath. Okay, some of you at school, all right, will use a Bunsen burner, all right, to keep it going. You guys must keep an eye on it, because what could happen is it's boiling water. If the camera shows you already, it's starting to go green, that it will go green shows that there is glucose present. So it will go from like a green to an orange, all right? But you must keep an eye on it. I have a wonderful pattern on my classroom roof at the moment, because some of my girls were not watching and the whole experiment exploded. So you need to be careful. It does make a good, interesting timepiece, all right? Talking piece on the ceiling, but you guys you must keep an eye on that if you're using the Bunsen burner. Okay, now let's get to our lipids. This one over here, all right, all I've put in is some usual sunflower oil, okay? And what I'm going to do, let me use the dropper over here, okay? See, little things so that you don't touch. All right, I'm just going to put water in there. Oh, and as you can see, if we can have a look, already you're going to see, look what happens. All right, the oil and the water are going to separate it. I'm going to just mix it, and then I'm going to leave it there to see what happens. Okay, now this is actually my experiment. That's a bit of my control. And what I have here is oil, but what I am putting into it is alcohol. Right, you can use ether, all right, and you can even use acetone. All right, those are the three things that you can use. And if you have a look, I think you'll see over there, already we don't see that emulsification happening. I'm going to just shake it, all right, for a while. There we go, you can see, and then I'm going to leave it. Okay, the next test that we're going to perform, all right, I think, cameraman, if you can just quickly turn over here, I want you to see, wow. All right, remember what I said about that turning orange? Okay, I'm sure now you have an awesome picture in your mind. Remember, it was blue, and now it's turning orange, so we can say that there's glucose there. Okay. So let's come back to the proteins. The protein test, there are two that you can use, all right? I'm using the easier one. Unfortunately, it is the more poisonous one. It's when we do proteins, our reagent, all right, is going to be Millen's reagent, which as you can see is white, or some of you might use, and I did bring some of it over here, okay, a combination. It's called the biuret test. You will see a combination of sodium hydroxide, right, which you make a solution of, and this lovely blue color that is copper sulfate. Okay, and if you make a solution of those, sodium hydroxide and copper sulfate, you can use it instead of Millen's. I just found it much easier to use with Millen's. If you're going to use Millen's, please remember just to use gloves, all right, because it is poisonous. Now, what are we looking at? I'm going to, if we have a look over here, I'm going to put some egg white. What are we testing for? To test for proteins. So I've taken some egg white, all right, and I have put it together with the Millen's reagent. Now remember what I said, it's poisonous, so I don't want it. Okay, I don't want to touch it. And again, I'm going to put it into the water bath, all right, because I want to speed up the reaction. Now, if we can come back to our lipids, I want you to have a look over here. What you will see, all right, this is our control. Remember, I took some water and I took some oil and I shook it up. And what do we notice? That the water and the oil are separate. Remember, water and oil, hydrophobic. They don't like each other very much. But if you have a look over here, all right, what do you notice at the top here? That the ether, all right, we've used alcohol, has dissolved. All right, it has dissolved. What do we know? Oil, one of the things that can dissolve it is ether. Now, it doesn't change color. When we look at lipids, it's not about a color change. All right, what I'm going to do is I'm going to pour a little bit, okay, just a little bit, onto there. There we go. And I'm just going to leave that. Okay, I'm sure all of you, all right, especially your clothes, and it's, I think, a mother's absolute nightmare, are going to find that when you put it onto filter paper, all right, you drain it onto filter paper, and if we just wave it a bit, what we're going to see, all right, not a color stain, but a translucent, greasy stain. All right, the water doesn't, it sinks into it. It's not like water that's going to make all of it. All right, runoff, if we look at it, we'll see that what does it do? It makes a greasy stain. Okay, so the last thing we're going to have to do, if we have a look over here, this is my piece of bread. All right, and all I'm going to use iodine. Now, if you have a look, I'm just going to put iodine, all right, there on the towel. What color do you notice iodine is? Excuse me. It is a brown. Okay, now remember what we said. Iodine solution, that's my reagent. And I'm going to be placing, wow, there we go, a few drops on the bread. Okay, what do you notice? An instant color change. This is iodine in its natural form, and this is what happens when it reacts with starch. It turns a blue-black color. All right, so if we were to recap, we've noticed that when I use Benedict's solution to test for glucose, a positive solution goes orange. Okay, when I test for lipids, my reagent that I use is ether or ethanol or acetone, and as you will see over here, what do I get? A translucent, there we go, you can see it, all these grease spots, all right? Translucent, greasy spot, not a color change there. If we look at starch, all right, what our reagent is iodine solution, and it turns a blue-black color. Okay, now have a look. The protein one is a lot or subtle. It's subtle when it comes into color. I want you to have a look there, okay? If you notice that, first of all, you'll notice that the protein went hard. Now, you guys must start to think. Proteins, you need to think of the properties of proteins. Heat denatures it. Very hot will denature it. So that's why my egg has cooked. But look, that is what we're looking for. Okay, when we use Millen's or Biuret, you're going to get a purple or a violet precipitate. Have a look over there. You can see it. All right, it changed color. We can put it over there. Okay, now why did I say that these food tests are so important? All right, remember our aim. We wanted to test to see if glucose, lipids, protein, starch, we had our apparatus, we had our method, and if they wanted to, in a practical, I could actually tabulate these answers. So what I could say is I could draw up a table and I could say test tube A, and I don't even have to know what's in it. All I can do is observe. It went orange. It made this. It went purple. It went blue back. Right? And then I can conclude from that there was glucose present, there was lipids, there was proteins, etc. Now this one, all right, guys, the reason why I stress about the protein one, please remember, enzymes are proteins. So the rules that apply to proteins also apply to enzymes. So we can use a protein test, all right, to use on any kind of enzyme test. And remember what I said to you. We're doing this to prepare you for grade 11 and grade 12. Remember, everything in the body is controlled by enzymes. So if we can do enzyme pracs, all right, it will, if you can keep in mind, positive reaction, violent. Okay, if we look at our glucose, you guys are going to do photosynthesis. You're going to do respiration. Glucose is either added or a byproduct. We can test that when we do the kidneys, when you grade 11, when you do excretion. If there's proteins in the urine, all right, if you test for proteins, there's something wrong with the person. If there's glucose in the urine, it could mean diabetes. So just by doing simple practicals, all right, you will be able to use these basic concepts that you will be able to use, all right, if you are given any kind of practical in the future. You might not necessarily, all right, have an idea. You might never have seen one before, but the basics apply. If these reactions happen, go back to your basics. Go back to if there's glucose, proteins, starch, and everything that's present there. So please, I hope you have a good, all right, memory, and keep these color changes in your mind. And when you get asked a question on food tests, that you're able to see the different colors, and it's much more visual for you. I think that's it for now. Katlego?
Thank you, Cheryl. So guys, we're going to go for an ad break. I know most of you are like, wow, this is so great. So now you know that you can actually have so much fun in life science. So don't move off your seats, guys, because we will be right back. Welcome back, Mindsetters. If you had joined us in the first segment, we had a food test practical, and it was amazing, guys. So in case you have just joined right now, make sure that all your friends are actually telling you what happened, because it was actually so great. And as Cheryl said, you will need it for next year and for the rest of this year. So I'm glad that you guys enjoyed that part. We're on to more theory now, hey?
Thank you, Katlego. Yes, guys, we're just going to look at a few questions that might pertain to practicals. But if you notice when I did the food test, there was really, really simple things that you could have used. If you didn't have test tubes, you could use little plastic beakers, you could use polystyrene cups. You need to think of what suits you. I think the most difficult things to obtain is obviously going to be your iodine and your biuret, your copper sulfate and sodium hydroxide. But I'm sure you guys are really, it's a really important test, and I do stress to you again, right, to get your teachers interested, to show you, so that you are able to have a good picture of those in your mind. Okay, now what I want to just quickly go on with here, when we look at it, remember we're looking at practicals, and when we look at a practical, right, it has certain components. It's called the scientific method. So when we do a practical, we need to do it in a way that is orderly and is set out. And as I was explaining to you while we were doing it, I tried to put in the terms like aim, all right? Hypothesis here was a bit difficult for the food test because you're not really sure what the color is going to be. If you already knew, then you could have stated hypothesis, all right? The glucose there would be lots of glucose in grape juice. All right, going on to that question, Katlego actually had two callers who asked, the questions were, what substance has the most glucose and what substance has the most starch? Now, that's a very tricky question there because you can't give a definitive answer. Right, just remember, what has lots of glucose in it? Sugars. All your sugars have lots of glucose in it. Especially, I use grape juice over there. If you notice, grape juice, why did I use it? Because it had glucose in it and quite a nice high content. Why didn't I use apple juice or any other kind of fruit? Because that has fructose, which is fruit sugar. All right, I could have also, to some certain extent, I could have also used milk because some milks also do have sugar in it, lactose. All right, so there's not a substance that's high in glucose, right? It's sugars. Any kind of sugars will have a high glucose content. And when you look at starch, I'm not quite sure what you mean by the amount of starch. Any starches have a high starch content, because what is starch? It's a whole lot of glucose, polysaccharide, that we put together. All right, so it's a whole lot of glucose put together, but we lose the sweetness as we did with our sugars. And our starches, rice, potatoes, bread, right? And the less they've been processed, the more their starch product, the more starch content they will have in it. Okay, I hope that answers your question, but I can't give you a definitive answer as to this has high, this has high. All right, so when we carry on over here, that was leading to hypothesis. Okay, remember hypothesis? This, I think, out of all of this, this is the most difficult question for you guys to think about. All right, remember it's a prediction. I predict that. I know you shouldn't say I predict that, but you think about it in your head. What do you think the answer will be? All right? And there's no but in there. I think the grape juice will go orange. All right? That is it. Not because the water was hot and the hot bath. That is, no, don't do that. Don't tell me reasons why. All right? Give me a prediction. All right? I think that the proteins will turn purple. That's it. I think that the proteins will not turn purple. I think that the egg white has protein. All of those are hypotheses. They are predictions. And remember, your hypothesis can be wrong, because at the end of your experiment, you're going to prove, is it right or is it wrong? All right, if your hypothesis was wrong, that doesn't mean that you were wrong. It just means you have to restart and relook at your experiment. Okay, we looked at apparatus, very important. Before I had to give you the show, what did I have to do? I had to make sure that I had everything in front of me, right? Because the much more organized it is, the much better the experiment is going to be, and also the much more reliable my experiment is going to be. Okay, this I also find the variables. This for me, I always have a chuckle, all right, at the kids, because these are the two things they battle with the most, the independent variable and the dependent variable. When you ask them the question, they seem to get it confused, yet when they draw the graph, all of a sudden the variable's on the right place. So I often say to them, draw the graph first and then answer your questions. Remember, independent. Independent starts with I, all right? They are the variables that I can change. All right, they're the variables that I can change. So in my food tests, what did I have control over? I chose to use grape juice. I could have used glucose powder. I could have used apple juice. Okay, I chose to use sunflower oil. I could have used peanuts. I could have crushed peanuts up rather than use the sunflower oil. So those are things that I can change. I chose to use egg white, right, instead of, for example, powdered milk, okay? I chose to use bread. I could have had a potato, I could have had cooked rice, any of those. Those are the independent variables. Those are the ones that remember, What can you choose? The dependent variables, those, I always say those are your results, okay? So what were my dependent variables here? My color changes. Right, when the glucose went orange, that was the result. When my protein went purple or violet, a little precipitate, that is my result. When my fat, my lipids, when they had a greasy spot, that is my result. All right, so when it comes to those things, very often you do tend to you get those two mixed up. Always remember, independent I. What could you change? What can I change? Okay. The fixed variables. When we get to a practical, all right, here not so much, but in other practicals, all right, certain things you want to be reliable. Certain things must be fixed. For example, if you are growing plants, all right, doing some kind of ecology practical, all right, you need to make sure that, like say, for example, I'm going to give you one, we acid rain. You set up little plot plants and you make up different sulfuric acid and you spray that. Right, you need to make sure that all your plants are in the same place. You can't have one plant here and one plant on this windowsill, one in the bathroom, because they're all in different environments. So your results are going to be, all right, not so true. Right, you need to, if it says, if you give it water, you must give that one 10 mils of water, that one 10 mils of water. You can't give one, all right, 10 mils and the other one 100 mils and expect your results, all right, to be reliant. So those are the fixed variables, the things that you need to do to contain your environment to make it, all right, as reliable as possible. Okay, then the method, that's set out exactly what you're going to do. You'll find when you do a practical test, it's the same as baking a cake. If you have a recipe in front of you, that is exactly what you do. You follow the instructions to have an outcome, and that's exactly what your method is. Right? Even not even seeing a practical before, just by following the instructions that you give you, step by step by step, that is what a practical is about. That's about practical skill, biological skill. All right, so that is conducting. Then you conduct the experiment, then the results. As I say to you over here, if I were to quickly put in here, I could have put a table in, all right, and I could have labeled color change at one heading, and I could have put test tube as another heading, and I could have labeled A, B, C, D, and then I could say there, A, what happened to it? It went orange, okay? So that is one of the ways, right? You're taking your data and then you're putting it into a table form, and then you interpret your data, all right? So what actually happened? Here, you can draw graphs. Some involve, if you've got a table, you can draw a graph. It involves numeric numbers, or just explaining what happened, all right? That is your interpretation. So why did my glucose why did it turn orange? Quite simple, all right? Because my grape juice, should I say, turned orange. Quite simple. Why? Because it had glucose in it, right? If I had used apple juice and it didn't work, then I would have said it didn't contain glucose. That's my interpretation, all right? And then the last one, right, is my conclusion, answering, all right, the aim. So to test for glucose, did I test? Yes, grape juice has glucose in. Okay, so if I was to carry on here quickly, as I said to you, please don't see the food tests in just one little mold. There are lots of questions, all right? Often practical elements of especially biology exam papers that you might have never seen the practical before. That's irrelevant. Look at what they are testing for. You could have soil. Remember last week when we looked at edaphic factors? Look at the soil, all right? You could do those little cylinders to see which soil drains water the quickest. Our food tests, enzymes, very important, all right? The enzyme question, I've got a question there for you. Osmosis. I don't know if lots of you did it at your school, but you could have done dialysis tubing, all right, with water inside and sugar water on the outside, or vice versa, where you showed the movement of water from a high concentration to a low concentration. Some of you might have used the potato. So lots of you, maybe, all right, you use the transpirational pull, all right? Sorry, let me just get here. Okay, when we get to transpirational pull, we use the potometer. Remember that air bubble that moved. So there's a lot of practical work, all right, that you might not necessarily do at your school, all right, do it practically, but that you definitely need the theory of it to answer the questions. If I look at questions, all right, this is just showing you again, all right, the food type. I just wanted to go over, very important. Know this. This is the most important thing you need to know. What was I testing for? What was my reagent? Benedict solution, iodine, ether, Millen's, or the copper sulfate. What was the normal color, and what did it change to? Have this. Make sure you don't need to know all the different food tests and everything. Know the table so that you are able to apply the question, as I'm going to show you in the next question over here. Here's an exam question. Now, before I even start to read the question to you, have a look at the picture. That's what I do first, all right? Have a look at the picture. And what do you notice? If we were to look there. What have we got? Oil, ether, we've got Millen's, and we've got Benedict solution, all right? So here, we're already looking at lipids, because we know our food tests. The Millen's reagent, what were we testing for? Proteins, okay, and Benedict solution, what was our testing for? Glucose. There we go. I haven't even answered the question yet, but what have I got a picture in my mind, all right, that it's the food tests. Okay, the question is, the following diagrams represent the apparatus used to investigate some organic nutrients. Study the diagrams and answer the questions that follow, okay? So if we go down, have a look over here. Test tubes 1 and 2 were shaken for a short while. These two, okay? 1 and 2 were shaken for a short while and then replaced on a test tube rack. What will be observed after each time? We've just done the food test. What do we know about oil and water? Okay, so there, answer your question. All right, what do we know about oil and water? Sorry. Okay, oil and water. Test tube 1, what will happen? They will separate. There's the word that I want. Okay, separate. What happens to the oil? Okay, the oil will be on top and the water will be at the bottom. But what happens to 2, all right? What happens to the oil in the ether? And the word there is dissolves. Okay? So it will separate the oil and water, remember, not firm favorites of each other. Okay? They will separate. You'll have the water at the bottom, the oil at the top. However, as we just look there, what did we find? That the ether is going to dissolve the lipids. Remember, that's one of its biological properties. Okay, the next question, right? Still on that. Okay, nice and easy. State the aim. State the aim of each of the following investigations. Number 3, what was the aim? Sorry, what was the aim for number 3? To test for proteins. Okay, nice and easy. Why? Because I saw my reagent. My reagent, right, identified what I was wanting to test for. Okay, then we go over here. Investigation number 4, Benedict solution. Right, what do we want to test for glucose. There we go. Right, nice and easy. Sorry, there we go. A little bit of a dead spot there. Okay, so just by looking at, look here, right, there's your food test. Okay, there's your food test. Able to answer those questions, all right, just by having now just watched the practicals. Okay, write the colors that will indicate positive results for investigation 3. What was investigation 3? Was my Millen's reagent. And what happened? What did it go? Purple or violet. That was the color change. Okay, and for investigation number 4, right, now hopefully you've got a picture when we use the word orange. Some of your teachers might use, all right, brick red. Okay, brick red. Sometimes that is also one of the colors that they use to explain it. Okay, so there we go. A nice question, right, based on the food test. And if you even had that block in your mind, right, of just the practicals thinking which reagent made which color, you would have been able to answer this question.
Cheryl, can we go to a quick break before you shoot over to the next one?
Oh, definitely.
Sure. So, guys, just before that long question, we are going to go for a break just so that you guys can get refreshed and you can get all your energy and your glucose levels up because there is going to be much more to tune into. See you just now. Welcome back, Mindsetters. As always, we have really short breaks here just so that you guys can get back to the important stuff. We are doing life sciences and we did a practical earlier. So now we're just going through questions that you can get on various practicals. So right now, if you have any questions whatsoever that you need to ask on what Cheryl has done today, please send them through right now because we are here to answer them. Over to you, Cheryl.
Thank you, Katlego. The next one we're going to answer is a bit of a long one, as you can see from here. But as I said to you, it's using the work, the theory that you have done, and this comes especially from the ecology section that you've done now, and trying to use that theory and put it into a practical sense. The question, I'm going to read it to you. It is quite a long one, all right? But have a look. When you're reading it, start already to think of, what is my aim? What is my hypothesis? All right, all of those things that we did in the scientific method. My aim, my hypothesis, what are my variables? What method am I going to use? How am I going to tabulate, etc.? What's going to be my conclusion? Those things already should be working through your mind. Let me read it through with you quickly. It says, decomposition is caused by micro and macroorganisms. I want to highlight that already. Micro and macroorganisms. So you need to know that the question is about decomposers. Right? Think about that occur freely in soil as well as inside organisms. A scientist wants to investigate the decomposition of leaves in 3 different cubes. All 3 cubes have the same volume. All right, same volume. What are we looking at? Our fixed variables. Let me go there. Our fixed... Oh, okay, that's not working. Okay, sorry. Let me just get to the eraser. Okay, now I'll leave that for now. All right, the same volume, okay, the same volume. That is one of my variables, and there's your volume, and are buried in the soil. So that is going to be my second variable. They're all buried, all right, together in the same soil. He designs an experiment to determine the percentage of decomposition in each cube. So we've got 3 cubes, all right, they all have the same amount of soil. They all have the same soil, all right, and that is so far is going to be your fixed variables. What he wants to know, all right, percentage of decomposition. What is that going to be? All right, that is going to be your results. That is going to be your dependent variable. He doesn't know what to, all right, expect. Now, what he does is the following. Okay, now this is where the difference comes in. Cube A consists of a wooden frame, okay, covered with porous material. In other words, okay, cotton material. So it's cotton, all right, and I want you to start thinking that it's got holes in it, okay? Then cube B is made of pine wood. So the whole thing is made of wood, all right? So it's solid, okay? But it still is wood. The last one is made of glass. So which of those two do you think would be the most impenetrable? All right, start thinking about that now. Now, when you look at your experiment, what are you looking at? You have got 3 different cubes. They're made out of different materials. So that is the only thing that is different about them. So the movement of the micro and macroorganisms into and out of the box is going to be dependent on these 3 cubes. Okay, so now formulate a hypothesis. You could have a variety over here. Formulate a hypothesis. Mustn't be a question, it must be a statement, and it must be a prediction. Okay, let me make one. I haven't seen the results yet. The results are below in the table, but I'm not going to use it. I'm just going to say, sorry. Okay, here we go. Formulate a hypothesis. More decomposition will occur, take a guess, in whichever one, in cube A. There we go. You could have said less decomposition, you could have said block cube A, B, or C. Right, remember what it is. It's just a prediction. What do you think the results are going to be? I've said more decomposition will occur in cube A. That's it, full stop. No reasons. I don't want to know reasons. I don't want to know but, because. All right, I don't want to know because there's this and that. That's it. All right, a nice, simple, short statement predicting what you think will happen. Okay, that is your hypothesis. If we go on to the next question, okay, you will notice, here we go. Okay, he obtains the following results. Here's the results. Remember what I said to you? One of the ways in which you can, how you put your results down is in table form. So now, what do we also know? One of our fixed variables is that he conducted the experiment on all 3 cubes, right, for 1 year. Not one month and the other one a different month, right? All three cubes, all under the same conditions. The only difference was, keep thinking in the back of your mind, the only difference was the covering of the cubes was different. So that must mean something. Now, if we were to look at our table, just by skimming up and down, what do I notice? My hypothesis was actually correct. Because if I have a look at percentage decomposition, what did I notice? Cube A, far more of the leaves decomposed than in B, and hardly anything decomposed in cube C. Remember? So what was in cube A? That was my cotton. All right, so that was material. Okay, and that was made of pine. All right, and the last one, cube, all right, that was made up of glass. Okay, so if we were to look at the question, here we go. Right, as I said to you before, what are the 2 things you need to be able to identify? All right, the independent and the dependent variable. Independent, I All right, I. What did you choose to use? What is your independent variable? The months. Okay, you chose to use it for the number of months. My dependent variable, all right, what was my percentage decomposition? I didn't know which box what was going to happen. What did I choose? Independent. I chose to do the study, or I chose to do the study for 1 year. All right, that was my choice. The dependent variable is what were the results? What could I not? What were the predictions? And those were going to be your percentage of decomposition. Okay, right, which will lead us on to the next question. Okay, use the results to draw a line graph, okay, that indicates all 3 curves of the cubes on the same set of axes. All right, 1 graph, 3 sets of line curves. Sorry, I'm having to extend the page here quickly. Okay, okay.
Do you have some?
There we go. All right, just needs a little bit of power, it seems. I need to have more protein in the morning. Okay, first thing that you do, all right, the basics of a graph. The first thing, I'm going to put it over here, a heading. Please, all right, I'm a bit pedantic about that. It's a line graph, all right, and now you must have your variables in it, showing the percentage of decomposition. There's my first variable. Of 3 boxes, shall I say, of 3 cubes over 12 months. All right, I've got all my variables. I'm telling you what graph it is. I'm giving you, here is my dependent, and here is my independent variable. Right, heading, very important. Now let's draw the axes. Okay, there we go. Right, we know it's going to be a line graph, and what do we put over here? The months of the year. January, February, March. I don't have space, let me put it over here. February, March, all the way through December. Okay, and what do we label it? We must label it. Okay, months. Right, oh, dead spot there. Okay. Then here, now remember, all of these should be the same distance. You can't decide to make this one 1 centimeter and that one 2. You always have a ruler, and each of those, all right, must be the same distance, the same as your y-axis. You can't make that 2 centimeters. They must all be, all right, exact. And let's see, we need to go up to 100. So let's go up in 20s, 20, 40, 60, 80, 100, and what do we need to add? All right, we must add a heading for it. So there we go. Before I've even started, 1 mark for my heading, 1 mark for my axes, labeling, 1 mark, 1 mark. I haven't even started to plot yet, and I already have 6 marks. The graphs are marks for jam, guys. If you know how to do your graphs, A and away for you, because they really are some easy marks to get. Now remember, okay, you had to put all 3 on the same set. So if you don't have different colors, all right, you can make 1 dot. I'm not going to plot it. You can make 1 that looks like that, okay. Then you make 1, then you can say that, remember, have a key over here and say cube A. Those are the ones with the line and the dot. Then you do all the dotted ones, and you say that cube B. There we go. All right. And the last one, you could maybe just make little dots. All right. Showing your graph. And that would be cube C, little dots. There we go. All of the 3 curves on the same set of axes. Remember what after our method, all right, what did we have? We had results. Those were results. What was the conclusion? The conclusion was taking the results and putting them into graph format. And once you put it into graph format, all right, what does it become a lot clearer? That there's going to be your graph A, right? If I have to do it, your graph A, which is your cube A, is going to have a much higher decomposition. So in which cube did more decomposition occur? In cube A. So it was the same as my hypothesis. So then I would be right. But I'm not finished now. Now I can answer, all right, questions on that. In which cube did the fastest decomposition occur? We can see over there, just looking, all right, at the results. Nice and quick. Let me see if I can. Okay. That would be number, there we go. All right, I need to, as I said, more power. In which, okay, cube number A. All right, let's go cube A. So nice, easy marks for jam. Okay, let's go to the next one. All right, those are just reading from your results, reading from your table. Now, this is the tricky question. Out of all these ones, this is why I put this one on. Why do you think the leaves in the cube mentioned in question 2 or 8.5, why did they decompose so quickly? Now, this is where your insight comes in. Everything else that you've done, all right, you've been able to get from the result. Now, I'm asking you why. Why do you think cube A, all right, had far more decomposition than cube B and C? And remember, let's go back to the first one. They all had the same soil. They all had the same amounts of soil. What was the difference? The covering. Okay, so the cotton covering, all right, the pine and the glass, that is going to be the factor that was the deciding factor. So what is it that cube A has got that B and C don't have? All right, and remember I said to you in the beginning, I said to you think of holes. All right, what does cotton have? Cotton, as I said to you, has holes. Right, and if you have holes, what can come through those holes? Aha, you're quite right. My micro and macro organisms. All right, so because they were free to pass into this cube from the soil, they could then decompose. Look at it again, watch cube A, the whole of its frame, all right, was porous. Porous means a material that can let things through. Pine, to a certain extent, yes, because it is a natural, all right, component. Some of them might have been able to drill holes through or to bore holes through, but you'll see that there's very little decomposition there because it might have taken too much time. And in cube C, nothing, glass. You know, they weren't going to be able to come through that. So, all right, have a look there. The cotton holes, all right, what did they do? The insects, the microorganisms, your bacteria, your decomposers that are so small, they could move in and they could move out. Okay, so I hope you're thinking about that first part when you're reading. Don't just read through the instructions when you get them. That little paragraph that you got in the beginning of the question, that was your most important resource, knowledge about the practical, even though you've never seen it before. Okay, our next one, I think it's a very simple question. All right, again, what percentage of leaves decomposed in cube B during the first 7 months? Okay, 7 months, all right, is in July. So if we have a look here, what percentage? Have a look over there in cube B. July is the 7 month. There we go. Nice and easy. 23. Do not forget your units of measurement, percent. And remember, look, it looked for cube number B. Read your questions carefully and read them thoroughly. Okay, I think we have time for one more. Okay.
Do you know, Cheryl, we can answer a few questions from the page.
I think that would, okay.
There was one by Lucinda who asked, they said that we shouldn't draw a broken line graph. We should draw the straight line graph, sorry. Would they lose marks?
No, she's quite right. All right, Lucinda, when I was just trying to show you the difference. What you could actually do is draw, when you draw, I'm going to draw in here quickly, hang on. When you draw your line graph, you do get marks for plotting points. Right, that your points must be joined. I would suggest, I must be honest with you, I would suggest that you use different colors. Right, but unfortunately not everybody goes into the exam with different colors. So if your teacher has told you to stay away from that, then do it. But then remember, when you go to an exam, you must have different colors. All right, I think that's
Okay,
enough for
that's
now.
it.
Thanks,
Thank you
Katlego.
very much, Cheryl. Thank you, Mindsetters. It's been such an awesome show. You guys love practicals. Some of you were saying that it's because you don't have to write so many notes. Maybe that's it. But I know that you also enjoyed it because practicals are always so much fun. So maybe you can try these at home as well. From us at Mindset, guys, thank you very much for joining us and goodbye.